Hi, I'm Park Eason, and I'm the NYP G9 science teacher at KIS International School, and I'm really happy to be able to teach the NYP science program because it's all about the wonders of science and teaching students about science skills that they'll find useful in the DP program and at university. In this regards, I think IB Science is one of the most challenging science curriculums out there, but it's also very rewarding for the students because in the IB system, students need to design, conduct, and analyze their own experiments. They have to use a lot of creative thinking and design and a lot of creative thinking in the analysis of that experiment, and that really sets IB Science apart from other science programs. In our current unit, Unit 3 of G9 Science, it's all about evolution by natural selection. And in this unit, there are two parts to the unit. The students learn about Charles Darwin and how he came to develop his theory of evolution by natural selections. Students also learn about modern scientists and the work they're doing with evolution by natural selection. And these two parts come together in the lab that we're doing in G9 Science, and that is a look at, does salt water kill seeds?
Okay, so guys, it's been a month since our seeds have been soaking, so let's make some observations.
I guess we'll see if Charles Darwin was right.
How does your water look this time, Sadie?
Mine is a lot darker than all yours.
Well, yes.
How does it smell?
It's always me. Yeah, how about no? Let's not make this into like some sort of perfume. It smells slightly like pumpkin, but not in a good way. Like it's not pumpkin soup. It smells like pounded pumpkin.
So do you think your seeds are going to germinate?
I'd say probably not because, well, obviously if you give salt water to plants, it's not going to be very good for their growth. And also because it's been here for almost a month, I think some of the salt has seeped into the seeds, so they're probably not going to germinate.
Yeah. What about you, Clara? They're so small.
Mine are mini seeds. I don't think that most of them are going to germinate. Maybe the ones at the very bottom will, but from my research, the ones that float are usually dead. And I have mint, and it doesn't do very well in high soil.
Yeah, same with mine, but all of mine have sunk, so I don't think they're going to do well either.
I think some of mine will germinate, but a lot worse than with the not salty water.
All right, ladies, so we finished the saltwater seed experiment, 28 days in salt water. We put them in the plant pot to germinate for 7 days, and we can see that some of the seeds did survive the salt water. We, as a class, planted over 2,000 seeds, and about 600 survived. And so here in the write-up to the experiment, we want to see why those 600 seeds survived and the others didn't. Is there a way that we can sort of divide the different seeds that we all planted to analyze what happened? All right? And I think together we can come up with lots of different ways. One of the simple ways to do it might be to take a look at which seeds are light colored, which seeds are dark colored, and then see if light seeds do better than dark seeds. That's not super scientific, and so I think you guys have some science that you might bring into yours to see what happens. So, Fa, tell me.
Well, you could start by dividing the seeds up by families in Genesis, because I think some families would probably be better adjusted to living near the sea or in places where there's more salt in the air like that, so you could try that.
Absolutely. I think when you divide up all the seeds, you're going to see we have about 15 different families of seeds. And we can, after we see the results from the 15 different families, we can see if one or two of those families is extremely good at tolerating salt. I think that one is excellent as well. So Molly, what do you think as well?
Maybe the size between big seeds and small seeds.
Yeah, we definitely looked at that. You can ask each of the students whether they think they have a big seed or a small seed and how well they germinated. Clara, what was your seed?
The lemon balm mint seed,
How many did you plant?
64.
And how many grew?
5.
And how big is your seed?
My seeds are really, really small. I could barely find them.
So side might be one of those things. Tell me, is there another way we can divide up the seeds?
Maybe by the seeds, like original climate. So if they like are native to North America or South America or Asia?
Absolutely. You'll see that we have seeds from all over the world, and maybe different regions do better, and then we can run the analysis on that. That's a great way of doing it as well. Sadie, what was your seed?
The winged bean,
How many did you plant?
20,
How many grew?
0.
So the winged bean, ooh, that's an interesting tie one as well. Other ways we can parse the data, we've got size, color, origin. All right.
We could look up at like the type of seed. So like some seeds might be like hard and able to break open, and others might not be.
That's right. We have this in science as well. We have two different types of seeds, the monocot seed versus the dicot seed. The monocot being the corn, that's only one seed, you can't break it apart. And the dicot, the beans, they all break into two pieces. And so there's a difference between monocots and dicots, and we could divide those things up. Lots of different ways to do this experiment, and I think that's what's really great about this. You have 2,000 data points, and as a group, you guys may want to choose which path to pursue for an answer. Yeah, excellent.